Welcome back to Student's Diary. Today we will discuss the first tutorial of Biotechnology course, that is Recombinant DNA Technology. This technology has laid the foundation of modern biotechnology. In the first step, we need a vector, or what we also call plasmid or biological vehicles, which will carry our DNA of interest into bacteria or any other suitable host. This plasmid contain different cutting site which will be cut by restriction endonucleases, an enzyme which is known as biological scissor. But remember, all these plasmid will be cut with same enzyme so that all plasmid cut at the same position. In the next step, we will cut our gene of interest which we want to clone. But remember that we will cut this gene with same restriction endonucleases with whom we had cut our vector. For example, our gene of interest is insulin. Once it is cut, now we will ligate this gene into this vector. Both genes and vector has been treated with same enzyme, hence it will produce same sticky ends. These both will incorporate to each other and form a phosphodiester bond. DNA ligase will be the enzyme which will ligate or seal this DNA fragment or gene of interest with vector. Now, the gene has been incorporated to vector. This is now called recombinant vector, or we also call that as recombinant plasmid because it contains the DNA of interest, or you can also say it contains the foreign DNA. As it contains the desired insulin gene, now we will look for a suitable host where we can incorporate this recombinant plasmid or vector, a host which is compatible to this recombinant plasmid. Most bacteria cells are used for this purpose. The process of introducing vector into bacterial cell is known as transformation. There will be hundreds or thousands of bacterial cells that may transform, but some may contain the recombinant plasmid which contain DNA of interest, in this case insulin gene, while other may contain non-recombinant plasmid and some may have not picked the plasmid at all. By using certain markers such as antibiotic resistance, we will select the desired clone which contain gene of interest. So we will get clone of our recombinant gene by simply multiplying those bacteria and plasmid inside it, and we will get required product. There are a lot of applications of recombinant DNA technology, such as the large-scale production of insulin, vaccine production for masses, production of insect resistance crops, and production of transgenic animal for the benefits of human being. So that was all brief introduction about recombinant DNA technology. See you in our next tutorial. Till then, Allah Hafiz.